Okay, we're going to kick off our second hour. We're going to have 4 speakers in this portion. And to kick it off, we have Wenjia Wang, who is a postdoc in the Huber Research Group in the Department of Chemical and Biological Engineering at UW-Madison. He specializes in chemical engineering and is passionate about the intersection of water, energy, food, and environmental sustainability. Please welcome Wenjia.
Good morning, everyone. I'm Wenjia Wang, chemical engineer, and today I would like to talk about what we did to utilize the pilot scale reaction and separation engineering to produce D Tagatose, low-calorie sweeteners from the Wisconsin cheese whey. So we know, as many of the previous speakers talked about, the dairy industry, particularly the cheese industry, is very important in Wisconsin. As the biggest cheese production in the U.S., Wisconsin produces about 1/4 of the cheese in the U.S. And a fun fact: for 1 kilogram of cheese you produce 9 kilograms of the cheese whey, which means in 2024, the state of Wisconsin produced more than 2 billion gallons of cheese whey. That is a high concentration organic wastewater. And what we know very interesting is that 5% of the cheese whey is lactose, disaccharide. Nobody likes it. So what we have done in the Huber's lab, we have developed a process to hydrolyze, use chemical catalytic process to hydrolyze the lactose into glucose and galactose, and we further use the chemical isomerization process to convert the galactose into a monosaccharide, a natural real sugar called Tagatose. It is an isomer of galactose with a very similar structure of glucose and galactose, but it has a very unique health benefit because it had only 1/3 of the calorie of the real natural sugar, but only has 92% of the sweetness of the sucrose. So that is FDA-approved natural sweeteners, alternative sweeteners, and it can be sold at $4 per pound. So that is something really interesting, and we have conducted a lot of work to use the pure lactose and pure galactose to produce the Tagatose. So there's one engineering question for our chemical engineers. So can we use a cheese whey, deuterated lactose, convert into a Tagatose-rich liquid, and we can use that for the downstream separation to actually produce the Tagatose powders. So, and we know Wisconsin cheese is a very good resource to do that work. So that is the objective of this study. So here I would like to briefly talk about the process we conduct here. So we firstly go through the feed conditioning process. We collect the cheese whey actually from the Babcock dairy plant. We go to different membrane filtration process to separate and enrich the lactose fraction of the cheese whey. Then we get the lactose-rich hydrolyzed feed. We do the lactose hydrolysis with the sulfuric acid as a catalyst, and we convert the lactose into glucose and galactose. Then we do the chemical isomerization with the calcium hydroxide to convert the galactose into Tagatose, and then we neutralize the calcium hydroxide to calcium sulfonate and go through different separation process to get a Tagatose-containing neutralized syrup, and we do different filtration and ion exchange resin to get a demineralized Tagatose-containing stream, and we use a process called SMB, simulated moving bed chromatography, to separate and enrich the Tagatose into a Tagatose-rich SMB extract stream that will be our feedstock for the downstream Tagatose powder production. So what we have done here, firstly, we conduct the membrane filtration to prepare a lactose-rich hydrolyzed feedstock for the actual reactions we did. That is really important part of our system. As we all know, the cheese whey contains different fractions: water, protein, fat, lactose, and those milk minerals. So we collect the cheese whey from the Babcock dairy plant. We did the ultrafiltration to cut the protein fractions, and we do the nanofiltration to concentrate the lactose fractions from 5% to 15 to 20%. Then we did the diafiltration to remove the some part of the inorganic burden fractions, particularly the chloride part, because at our elevated hydrolysis conditions, those chlorine ions can be corrosion, cause serious problem for our system. So we have done the batch top filtration at the Professor Gustan's groups at 5 gallons, and we scale up the system to conduct at the Center of Dairy Research. In this February, we conduct 250 gallons of the cheese whey to get the final protein-free, lactose concentrate, chlorine-free feedstock. As you can see, after 3 steps of the filtration, we concentrate the lactose from about 5% to 15% and remove the protein and remove the most of the chlorine ions. So that is conditioned as a very good feedstock for our next step, hydrolysis. And after that, we conduct the lactose hydrolysis in both batch system and the lab scale continuous flow reactors. So the lactose hydrolysis process is highly affected by the conditions of the temperature, pH, and the reaction times, and it will build up kinetics windows for how we operate the lactose hydrolysis in the next steps. So we conduct the kinetics studies to build the kinetic models to predict at what temperature and time and the pH we could hydrolyze those lactose into galactose and glucose with a very quick reaction rate and a very high yield. We also focus on the formation of those unwanted byproducts, the degradation products of those lactose hydrolysis. So it's kind of help us to guide, determine what will be the severity windows of the lactose hydrolysis, and these kinetics models have been verified in our batch lab scale system and in our batch scale lab scale continuous flow reactors at 5 milliliter per minute. So that kind of figure out the chemistry and the kinetics operation windows of the lactose hydrolysis. And what we did next step is scale up the system to the actual pilot scale. So we built up our pilot scale rack system at the Babcock dairy plant, and it can be operated at 1 gallon per minute at the determined hydrolysis systems. So when you operate the large-scale system, it's no longer an ideal batch system at small scale. You should consider all the mixing, all the space and space distribution and thermal history explosion of your system. So as you can see, when we integrate our kinetics model from our batch to the large scale, as you can see from the green lights, it may not match well with our measured lactose measured products concentration profile. But if we consider those take-up space of the system, so in the first almost 30 minutes, those we permit just need to fill up the whole system. So at the outlet of the reaction, you actually didn't see the hydrolysis reactions happens, but after 30 minutes, we saw those hydrolyzed lactose formed, and those glucose-galactose and all those byproducts forms. And also, because that is a very large reaction system with a total volume over 30 gallons, the thermal explosion history also matters. So we developed a different thermal history modeling to match our output method. So it's kind of like both the space distribution or take-up of your reactors and the thermal history of the lactose hydrolysis really matters when you want to scale up your system. And with this system, we processed over 300 gallons of the cheese whey per minute, and we produce those galactose glucose syrup with very high yield over 90%, and we produced maybe 150 gallons of those feedstock. And those feedstock will be the next step feedstock for producing the actual Tagatose. So what we have discovered in our lab is we can use the very rich chemical reagents, calcium hydroxide, to isomerize the galactose into Tagatose. When we use the galactose glucose syrup as a feedstock, there are multiple reactions happens. You have the conversion between the galactose and our targeted products, Tagatose, also from some byproducts are called tallose. It's also a health benefit, low-calorie sweeteners. You also will have the isomerized from the glucose to fructose. That is unwantedly reactions, but you can control it with turning the conditions to play with the kinetics and the thermodynamics. So what we have seen from our HPLC profile after the galactose glucose syrup isomerization from the actual cheese whey, you can see we formed the Tagatose, we formed the lactose and other monosaccharide, and we still have some fractions of the lactose. So what we have produced from the lactose hydrolysis and the cheese whey hydrolysis is actually the mixture of most of our targeted products, Tagatose, but also have another monosaccharide and disaccharide, those sugars, and you also have some organic impurities and the inorganic burdens. So what we need to do is to purify that stream. That is come to the third process of our system. So yeah, so we did the sulfuric acid neutralization and to neutralize the calcium hydroxide to calcium sulfonate. And after that, we did the filtration to remove both the calcium solid. But during the filtration systems, we can see from here, so after the neutralization, you formed a calcium sulfonate cake, a slurry, and you did the filtration, you can separate most of the sugars into the filternate, but you also form the calcium sulfonate wet cake, and it will hold up the sugars in the wet cake. So you need input more waters to wash the cake and further separate the holded sugars into the washy solution, and finally you can get the mostly sugar-free cake. So as you can see from these here, the figures here, so before the isomerization, we probably have 240 grams of the total solid, about 210 grams of the sugars, but after the first filtration, you only recovered maybe 70% of the sugar from into the filternate, and you need another washer to transfer those sugars into your final products. So those sugars are recoverable, but also you need input more waters, and it also increase the downstream burdens of your system. So it's kind of a trade-off to how can we balance the water input and the downstream operation cost, as well as how much the sugar you can. Yeah. So yeah, and we also did the deionization works to use ion exchange resins to remove the sugars, but also while you deionize the streams, you also hold up the sugars in your resins, which causes another trade-off between the operation cost and your output. So yeah. So, and after that, we use a simulated moving bed to enrich the streams, and we can enrich about 32% lactose into 70% lactose. This is also our preliminary data from 50 gallons of the lactose containing streams. And yeah, so we are still a lot of things ongoing to do. So what we know is we demonstrate this process can actually use the actual cheese whey to do the lactose, and we need to do more research about to scale up the whole system. Yeah. And I want to thank my PI, my agents, and all my undergrads and collaborators. Thank you.
One quick question.
I was just wondering, you split the lactose, you get galactose and glucose.
Yeah.
So in your waste stream, you will have glucose.
The glucose will be recycled to do the biofermentation. Yeah. Yeah.
All right, thank you.